Okay. So good morning, everyone. Please answer your attendances. Roll number, roll number 766,
Yes, sir.
67, 68,
Yes, sir.
68, 69,
Yes, sir.
70,
Present, sir.
71,
Present, sir.
72,
Present.
73, 74,
Present, sir.
75,
Present, sir.
76,
Present, sir.
77, 78, 78, 79,
Present, sir.
80,
Present, sir.
81,
Present, sir.
82,
Present, sir.
83,
Yes, sir.
84,
Present, sir.
85,
Present, sir.
86,
Present, sir.
87,
Present, sir.
88,
Present, sir.
89,
Present, sir.
90,
Present, sir.
91, 92, 93, 94,
Present, sir.
95,
Present, sir.
96,
Present, sir.
97, 98, 99, 800,
Present, sir.
1,
Present, sir.
801.
Yes, sir.
आपका नाम कार्तिका पटेल है.
कार्तिक पटेल, sir. हाँ.
Okay. 802,
Present, sir.
3,
Present, sir.
4,
Present, sir.
5, 6,
Present, sir.
7,
Present, sir.
8,
Present, sir.
10,
Present, sir.
11, 12, 13,
Present, sir.
14, 15,
Present, sir.
16,
Present, sir.
17,
Present, sir.
18,
Present, sir.
19,
Present, sir.
21,
Present, sir.
22,
Present, sir.
23,
Present, sir.
24, 25,
Present, sir.
26,
Present, sir.
27,
Present, sir.
28,
Present, sir.
29,
Present, sir.
30,
Present, sir.
31,
Present, sir.
32, 33,
Present, sir.
34 is not there, 35, 36,
Present, sir.
38,
Present, sir.
39,
Present, sir.
40,
Present, sir.
41,
Present, sir.
42,
Present, sir.
43, 44,
Present, sir.
45,
Present, sir.
46, 47,
Present, sir.
48,
Present, sir.
49, 50.
Present, sir.
Okay, so in the last class we were studying about
Sir,
yes,
sir, it more present.
8, 11%. 8 minutes, okay. 811. Okay. Anyone else? Yeah, everyone, is my screen visible to you all?
Yes, sir.
Everyone, is my screen visible to you all?
Yes,
Yes,
visible.
sir.
Yes,
Yes,
sir.
sir.
Yes, sir.
Yes, sir.
Yes. Okay, so in the last class we were studying about the formation of a DNA chain. Okay, so let's just have a quick recap of what we have learned earlier. So we have seen that we have two different types of chains in the nature. One is the DNA and the other one is RNA. So DNA usually will be forming your genetic content and RNA is the supporting content that is required. But in certain cases, RNA also can form the genetic content as we have seen in case of viruses. Okay, but majority of the creatures on the earth have DNA as the genetic content. So when we look at the formation of DNA and RNA, we have three important structures to be studied. Number one is your nitrogen base, number two is your sugar, and number three is your phosphate. Okay, and phosphate is a high energy molecule which will be crucial for the establishment of these bases. Okay, so when we come to nitrogen bases, we have two different kinds of nitrogen bases. We have purines and pyrimidines. In purines, we have adenine and guanine. In pyrimidines, we have thymine, cytosine, uracil. Okay, so in DNA we have adenine, guanine, thymine, cytosine. These four. And in pyrimids, we have adenine, guanine, cytosine, and uracil. The difference between uracil and thymine is that in thymine we have an additional methyl group. With respect to uracil, if you are going to see, we have an additional methyl group in thymine. That's the only difference. So this occupies DNA and this occupies RNA. Now coming to the sugar, we have two forms of sugar. One is deoxyribose and the other one is ribose. This is the regular form of the sugar that is available in the nature. And this is the modified version of the ribose where we are lacking a oxygen at the second carbon, and hence we call it as 2-deoxyribose. So if this goes into the nucleoside formation, nucleoside क्या होता है? Combination of a sugar and a base. If ribose goes into the nucleoside formation, it's RNA, and if deoxyribose goes, it's DNA. Simple as that. Now comes the third factor called as phosphate. So how does the phosphate bind? Through a ester linkage. Okay, we have a phosphate and we have an oxygen here. Okay, so oxygen of the phosphate tends to interact with the fifth carbon of the sugar, and that forms something called as a nucleotide. Okay, nucleotide में क्या है? We have a base, we have a sugar, and we have a phosphate. So this three combination of three is called as nucleotide. So now once the nucleotides are formed, then goes your formation of the chains. So how does the chain happen? The same phosphate will tend to interact with the OH present on the third carbon, and this creates another linkage, ester linkage. So this two bonds that we are seeing here together can be called as phosphodiester bond. Okay, so through phosphodiester bonds, we are seeing that the DNA chain is forming. So whenever your DNA chain is forming, remember the interacting partners are always 5 and 3 of two different sugars. It starts at 5 and ends at 3. Starts at 5 and ends at 3. So this is the direction of synthesis of your DNA chain or an RNA chain. Okay. So we always determine the direction as 5 prime to 3 prime. Whenever you are indicating any DNA molecule or RNA molecule, you have to indicate this priming. Okay, just to indicate the direction of the uh, what do you say, the direction of the chain. Now, when we are talking about this is where we are running. Okay, so now continuing with this, when this DNA chain has to form, and when the double helical structure has to form, these so-called bases now goes for interaction. So when they have to interact, there are different concepts that we have to understand. Number one, the interaction should be thermodynamically feasible, and number two, for the feasibility to happen, the sugars are never in the parallel direction. They are in the opposite direction. Example, look at this figure or look at this image here. You can see the direction of the sugar here and you can see the direction of the sugar here. Okay. So if your 5 prime end is here, here we have a 3 prime end. Okay. So the orientation of the sugars of the interacting partners are opposite always. Number one. Number two, while and coming to the interacting partners, it is observed that purines never interact with purines, pyrimidines never interact with pyrimidines. Matlab, A never interact with G and C never interacts with T. Okay, so that's ruled out. Purine and purine interaction, pyrimidine and pyrimidine interaction is ruled out. Now let's look at purine and pyrimidine interactions. We have A, T, G, C. A and C are not thermodynamically feasible. Okay, A and C are not feasible at all. At the same time, G and T are not feasible at all. So the only remaining partners are A and T. A stands for adenine, T stands for thymine. So adenine goes for it, bonding with thymine, and it will be able to form two hydrogen bonds, as you can see in this figure. Okay, and guanine and cytosine forms three hydrogen bonds. So this way, these are the only true interacting combinations that you see in DNA. Okay, I am saying in DNA. When it comes to RNA, we have a T to be replaced by uracil. Okay, so this way adenine will be interacting with thymine in DNA or uracil in RNA, and guanine will be interacting with cytosine only. Okay, so it's ATGC. Remember, AT have two hydrogen bonds, GC have three hydrogen bonds, and this way when we show the structure of the DNA, we see it like this. We show all the base pairs. We are saying base pairs, which means we have what interactions happen between these particular nucleotides. Okay, so this is a pair, this is a pair, this is a pair. So this way we have different base pairs, right? That's number one. So when the base pairing happens, always remember the orientation of the sugars are opposite. They are not same. So you don't have a parallel chain of DNA. We have an anti-parallel chain of a DNA. Okay, so this is how you can see one chain synthesis has happened from 5 prime to 3 prime manner, and the other one has happened in a reverse manner. Okay, so this is the structure of your DNA. Now, coming to the combinations, like we have, look at the combination of interacting partners. There are only two possible combinations: is AT, adenine, thymine, and guanine, cytosine. Okay, so depending upon this, can identify like if we come to know about one particular base content in a particular DNA molecule, you will be able to identify the rest of them. Okay, how is it possible? Through a rule called as Chargaff rule. Okay, which says that 1, 2, 3, 4. We have four bases of DNA. All the four bases together is 100%. Okay, all the four bases together is 100%. Let's say we know about the base adenine. Okay, let's say its content is 20%. Okay, so adenine 20% है, so automatically thymine भी 20% होगा because this is the only binding partner of adenine in DNA. Okay, so straight away it's going to be 20 by 20 20. So here we have 20 and here we have 20. So 40 is occupied by 80. So remaining कितना है? 60% remaining है. 60 में both of the share equally. 30 will be shared by guanine and 30 will be shared by cytosine. Okay. So this way, if you come to know about one particular base, automatically you will be able to recognize rest of the other bases also. Okay, same way. Let's say cytogen is 25%. So cytogen 25% है, so automatically go on and be 25% होगा. Okay, so 25 plus 25, this is 50, and we are left with another 50. So that another 50, again do the sharing 25 and 25. So this way we can identify if you come to know one base automatically applying the base pairing concepts, you will be able to understand or you will be able to understand the percentages of other bases. Okay, so this way, let me just give you a problem. This way, if our adenine content is, let's say, 13%, what will be the content of T, G, and C? Answers on the chat box. Do the calculation and write your answers on the chat box. Yeah, answers are right. Yeah, answers are right. T is 13%, G is 37, 37. So GC will be 37, T is 13%. Good. Now, moving ahead with the concepts again. Now let's look at the pyrimidines and purines. Okay. Let's look at the pyrimidines and purines. We have pyrimidine as adenine, thymine and cytosine. These two are the pyrimidines and these two are the purines. Okay. So हम जब ये दोनों का और ये दोनों का रेशियो लेंगे, it will be every time 1. Okay. Look at the same calculation that I gave you. Adenine का content कितना था 13 था and guanine का content कितना था it is 37. Calculate both adenine and guanine 13 plus 37 is going to be 50. Same way calculate thymine and cytosine 13 plus 37 50. So the ratio of purine and the ratio of pyrimidine will be equal to 1. Okay, that's another way of determining this role. The ratio of pyrimidines and the ratio of purines is equal to 1. Okay, we get 50% of purines or 50% of pyrimidines in a DNA molecule, and that's true. Okay, because theoretically we get 50%, but technically when we conduct the experiment and when we calculate all the bases in the entire DNA stage, okay, if not 50%, at least we will be getting 49.99 at least. Okay, so that's again equivalent to 50. Okay, so we get pretty close values experimentally as well, and that's the reason why people do follow Chargaff rule even to date. Okay, so remember the role. The role name is Chargaff. Okay, he's a gentleman who discovered the nitrogen bases contents in a DNA molecule. Okay, and based on him only, we are doing all these calculations. Okay, so this यहाँ तक बता दीजिए. Do you guys have any difficulty in understanding till here? Is the concept of the basis and all clear?
Yes,
Everyone?
sir. Yes, sir.
Okay, okay. Now let's go ahead with the next molecule. Okay. So the other molecule that you come across is RNA. Okay, so RNA, we have a formation of RNA along with the DNA in the nature. Okay, it's not like DNA start ho gaya hai and RNA की जरूरत बिल्कुल जरूरी नहीं है. It's never like that. Okay, in nature, when the nature has constructed DNA was there, and because of its too many hydrogen interactions, the DNA tends to be much stable. It stays for a very long time. Okay, hydrogen bond, of course, is weaker, but when you look at the DNA molecule, you have some millions or billions of bases available in a DNA molecule. Okay, this means that you have billions of bases into the number of hydrogen interactions. You can see either two interactions or three interactions. So altogether, if you're going to look at the interactions, probably they will be again in billions of hydrogen interactions. Okay, so a single molecule with these many number of hydrogen interactions is again pretty strong, very strong to break. Okay, so you require enzymatic catalysts to do the breaking process. I say he straight away they can't break. Okay, so that's the reason. That's the reason why you see DNA molecules even till date. Let's say we are getting some fossils of around like 5,000 years or 10,000 years. किसी को कुछ fossil मिल गया है. So from the fossil, they will be able to extract the DNA and they will be able to identify the DNA and then do the studies on it. Okay, this is like one of the reason why to determine its stability. Okay, whereas RNA molecule is simple standard. It's only single stranded. Majority of the times, single stranded, and this RNA molecule, as it is single stranded, and as it doesn't have much of the bonding and all, and as you have a ribose sugar in it, okay, it gets easily degraded compared to a DNA. Okay, and that's the reason why you don't see RNA available in most of the decomposing creatures or most of the fossilized creatures. Okay, so however, we have requirement of RNA in all the living forms. Okay, as far as we are living, you see the RNA. So where do we see RNA, and what are the kinds of the RNA we see? We see RNA in three formats. One is messenger RNA, number two is ribosomal RNA, number three is transfer RNA. Let's learn about messenger RNA. Now we have a DNA. The DNA has genes. Let's say we have 10,000 genes on the DNA. Okay. Let's say now we are getting some sort of a uh, changes in the weather. All of a sudden, let's say rain occurred. Okay, कहीं ना कहीं, let's say you're passing through some street and then अचानक से बारिश हो गया है. So when the rain when it's raining, your skin cells and your entire body will be giving the stimulus to the internal cells, right? So the stimulus has to be passed on, and we have to react, right? So your muscles, your musculoskeletal system, in association with your nervous system, will be allowing you to move to a separate location where it's not raining. Okay, that's one thing. Second thing, as we have some water content on our cells and all, okay, and as there is a change in the temperature, exterior temperature, so our skin cells and our muscle cells and our internal cells has to what do you say, get back to the normal condition. To do that, whatever the stimulus we have received has to get passed on to the genes. So let's say now we have 10,000 genes, and let's say out of this 10,000 genes, let's say 30 genes has to get activated to perform this activity. Okay. So 30 genes activate होने के लिए, now you see the number of the genes on the DNA are 10,000 and we hardly require 30. So you can't every time pull the entire DNA out and then you can do the work. That's that's quite impossible. And DNA is not a small molecule; it's a very big molecule. And if you're going to look at the what do you say, availability of the DNA, it's available as double chain format. Okay, so working on the DNA, especially with the double chain format, is again a tedious task. Okay, well, in a layman's language, let me tell you. Let's say we have Ankit here, and let's say we have Neha here, and let's say consider myself, three of us. Okay, Ankit, Neha, and myself. Let's say Neha is the protein which is acting as a transporter. Let's say Ankit is your RNA, and let's say myself is DNA. Okay, I am the DNA. Ankit is RNA. Neha is the protein transporter. Okay, Consider RNA as 50 kg and consider myself as 120 kg. Okay. I mean, we're talking about a specific gene, only gene, not the entire DNA. एक gene का weight समझ लीजिए. So uh, in terms of human language, we are just saying. So मेरा वजन 120 किलो है, अंकित का वजन 50 किलो है, and Neha is the transporter who has to drag the RNA molecule or the DNA molecule out of the nucleus into the cytoplasm. Okay. So when Neha is working on Ankit, or transporting Ankit, and when Neha is transporting myself, where is Neha going to spend more amount of energy? Yeah, there's a question to every one of you. When Neha is pulling me outward out of the nucleus, and when Neha is pulling Ankit outward of the nucleus, कहाँ पे नेहा ज्यादा एनर्जी स्पेंड कर रही है?
120 kg.
So
Yeah.
120 kg on you, sir.
120 kg.
It will be
Yes.
120 kgs. It will be on me. Okay. Simple. Just for understanding, I'm saying. Okay. So you have to see when we are looking at the cellular thermodynamics, majority of the energy utilization will be in a conserved manner. It is not going to be in an expended manner. It will be in an energy conserved manner. Our cells are evolved to be functioning like that. Okay. So हम इतना एक particular gene के लिए you can see the difference what the protein transporter is undergoing through. Okay, so RNA molecule is pretty simple. It is small and it can easily pass through all the sorts of what do you say passages. Whereas DNA molecule, it's double. Number one, it has many other extra elements in it, and transporting this DNA molecule is again a tedious task. Okay, so in this manner, when we are transporting a DNA and an RNA is the choice for majority of the transporters. So what we do is that हमारा ये जो gene है, whatever the gene we have, so the same gene, we will be making it into a small copy in the form of an RNA. Okay, and this RNA is carrying the message of the same gene, and hence we call it as messenger RNA. Okay, whatever the codes are available on the gene, only these codes will be taken up by the RNA. Okay, and hence we are calling it as messenger RNA. And remember, messenger RNA is only available whenever the genes has to get activated. Okay, it is not available throughout the cellular course. Okay, so for this reason, messenger RNA is the least content of RNA available in the cell. Okay, it hardly produce it is hardly it hardly makes around like 5% of the cell, entire RNA in the cell. So this is one format of RNA. Okay, where your DNA has to get converted into RNA and the processing further will be happening only on the RNA. So this copy of RNA is called as messenger RNA. This is number one. Number two, ribosomes. We have studied about them earlier in differentiation between prokaryotic cell and eukaryotic cell. So we have 70s type and we have 80s type. So both the types usually have two subunits. We have a small subunit and we have large subunit. So the subunits are usually made up of RNA. So what does this ribosomal RNA do? It takes parts in providing all the requirements for the protein synthesis. Okay, ये जो molecule यहाँ पे आप देख रहे हैं, this chain that you are seeing here is this single stranded. This is your RNA. Okay, from your RNA we have this bases. These are your nitrogen bases. Okay, so these three nitrogen bases will make one code, and based on this, you will be getting this amino acid chain. Okay, so the process is called as translation, which means we are decoding the message and we are getting a product. Okay, and that happens on your ribosomes. So if you're going to look at the cellular RNA, majority of the RNA is available in these structures. Okay, more than 80% of the RNA is in these structures. What are these structures called as ribosomes? Okay, so the RNA available in the ribosome is called as ribosomal RNA, shortly abbreviated as rRNA. Okay, then comes the third format of RNA called as tRNA. T stands for transfer. So what does it do? When your DNA is synthesizing messenger RNA has some codes, right? As I said earlier, you can see these bases here. So the bases will be doing the coding in triplicates. Okay, triplicate matlab every three bases will make a code, and this code is the thing that has to be decoded. For example, let's say we have C U U. For example, let's say we have C U U on this RNA as a code. Okay, so when you go to tRNA, you can see tRNA has the complementary. We have three loops: one D loop, T loop, and anticodon loop. So anticodon loop is very crucial for recognition of this code. So what we have seen C U U here. So automatically उसका complementary क्या होता है? G A A. Complementary means like pairing partner. Okay. So C binds to G, U binds to A, and another U also binds to A. Okay. So this is RNA we are talking about. This is not the DNA. Remember, this is not the DNA. This is RNA. And hence, you call this kind of a loop as anticodon loop. Okay. So we have a DNA with cloverleaf model. We have a stalk. The stalk at the 3' end bears the amino acid, and this amino acid is dependent upon the uh, anticodon available here. Okay, so this anticodon is the interacting partner with your RNA, and we have different loops called as T loops and D loops. These are regulatory loops that are required for the protein synthesis that takes place on the ribosomes. Okay, so this is a cloverleaf model which says that we have a stalk and we have a crown. So your tRNA usually will be looking like a clove. Okay, clove is kind of as a species spice that we use in majority of our dishes. Okay, in Hindi we call it as long. So this is the structure of your tRNA. So we will be studying about these loops in detail in protein synthesis part. As of now, it is not required. Okay, and coming to the content of tRNA, it's approximately 15%. So if you're going to look at all the three formats of RNA, we have mRNA, tRNA, rRNA. rRNA is the highest format, crossing more than 80%. Then tRNA comes next, it's 15%. Then mRNA is the least, probably it makes hardly 5%. Okay, so these are the three different types of RNA that we come across in the nature, and these are the different functions. They are required for making of proteins. tRNA they are required for decoding process. mRNA they are required for transferring the codes from DNA onto the simple molecule of RNA. Okay, so this way all the three has different functions. Ribosomal RNA, this is a much abundant form of RNA. This is available everywhere. You find them on the nucleus, find them inside the nucleus, you find them on the endoplasmic reticulum, you find them freely available in the entire cytoplasm. Okay, same with usually tRNA is associated with ribosomes because the ribosomes have all the protein synthesis machinery, and mRNA it forms in the nucleus, it comes out of the nucleus, gets uh, takes a position in the ribosome. After the decoding process, it gets back and gets recycled. Okay, so this way we have all the three formats of RNA in the nature. Now, kill RNA. If anyone has any doubts, let me know. Kill
Excuse
RNA.
me, sir.
Yes, yes, yes.
Sir, for mid-semester exam, hello?
Yeah. Tell me.
Yes, sir, for mid-semester exam, do we need to study only the presentation prepared by you or we need to study the book also, sir?
Book is better, but whenever I say you have to read this from the presentation, that you mark because there are certain contents in the book which are not available, like microorganisms is not there in the books. So you have to read the content from the slides. The rest of the content you can prefer book.
Okay, sir, thank you, sir.
Book is the better option because in presentation I am using it for explanation purpose. So you may not find every content on the presentation, especially the notes that you will be looking for. You won't find it on the presentation. You find figures. Okay, I'm just using this for explanation purpose. Okay, majority of the notes you find it on the textbook only.
Okay, sir.
Okay, so RNA में dots किसी को कोई doubt है RNA में?
No, sir.
No. Okay, we'll go ahead then. Now, moving ahead. So we have our DNA, fine. The DNA we have studied that when the cell divides and reproduces, the DNA gets transported from one cell to other cell. Okay. So when a cell is dividing, you see, we are using the term called as division, which means from a single cell we are getting more number of cells. Okay. So when we get more number of cells, how does this single DNA get into two different cells? Okay, so that's one thing. Second thing, if you're going to look at the DNA is double helical in nature, and if it's going to stay as such in the body or in the cell, it's going to occupy a huge amount of space. Okay, example, consider our DNA. Among our DNA, we have some trillions of cells in our body, and we have some megabases of DNA available in our body. Okay, so if you're going to look at these megabases of DNA, and if you're going to look at this trillion number of cells uh, with the DNA available in our body, sorry, not megabases, probably it's more than 10 power 9. So with the content of the DNA in our body, you can actually, if you're going to stretch the DNA, you can actually go to sun and get back to the earth like four times, not even like one time. It's four times. Okay, so you can tour around sun and get back to the earth four times. So that's the length of the DNA that we have in us, in all the cells together cumulatively. Okay. So if you just leave the DNA like that, probably it's going to occupy the more space. It is not going to fit in this 2 meter body. Okay. So how is this DNA then fit? कैसे fit हो गया है? Especially how is it fitting in these microscopic cells? Okay. So it undergoes compaction. Okay. Like when you have a larger file, you will be using a lot of compressing tools, right? And you will be compressing the file like a MB file to KB, and you compress it like more than 1,000 times, or even greater than that. The same way we are compressing the DNA using different protein molecules. We call them as histones and proteins. Okay, and we are converting them into structures called as chromatin. Okay, and from the chromatin, we get robust, linearized, rope like structures called as chromosomes. Okay, so chromosomes, what does the chromosome mean? Chromo is something that takes the stain, color, and soma means body. So a body which can actually take more color is called as a chromosome. Okay, this was observed by a gentleman who was actually studying the salamander larvae. So when he saw the cells dividing, he has seen that these colored bodies are moving from one particular cell to the dividing partners. Okay, so his name is Walter Fleming. And when you look at the chromosome numbers, we have two conditions. One is haploid and the other one is diploid. Haploid क्या होता है? If you have a single chromosome, let's say you have in humans we have 46 chromosomes, as we can see here. Okay, so the 46 chromosomes are actually not the original chromosomes; they are in pairs, they are in couples. Okay, so we have 23 pairs of chromosomes. You have to see like that. We have 23 pairs of chromosomes. So this 23 number, some of the cells can have only 23 number. So that's actually one set of your chromosome. So one set is called as haploid. Haploid stands for one set of chromosomes. And if you have two sets, you call it as diploid. If you have three sets, we call it as diploid. So in nature, we usually come across either a haploid or a diploid. In cases of plants, you can even look at polyploidy conditions. Polyploidy means more than two. Okay, we can see triploidy, we can see tetraploidy, and so on. Okay, so we have different kinds of ploidy. Ploidy, Ploid, determines the number of sets n of the chromosomes available in that organism. Okay, so this way, if you are going to look at the diploid numbers, two sets of chromosomes, human beings have 23 pairs, onions have 8 pairs, cats has 19 pairs, corn has 10 pairs. Okay, so chromosome का formation कैसे होता है? Let's look at the packing. So when the DNA has to pack, first it has to interact with the proteins called as histones. Okay, histones. Remember the name of the proteins. So in histones we have five different types: H1, 2A, 2B, 3, 4. Okay. So H1 is separate. 2A, 2B, 3, and 4 in multiples of 2 will be making an octomeric complex. Octomer, 8 units. And 8 units are made by 2 units of 2A, 2 units of 2B, 2 units of 3, 2 units of 4. Okay. So this makes this octomeric complex. So this is going to act just like the way we have our thread roll. हमारा जो धागा का roll है. Let's say this is going to act as the base of the roll. And let's say our DNA is our dhaga. Okay. So what we do here, we bind our DNA across this octomeric complex. Okay. So this complex with the DNA is now called as a nucleosome. Okay. So we can see we have a one nucleosome formation here. The DNA is wrapped around this octomeric complex. So we have one here, one here, one here, one here. So this way we have different nucleosomes complex being formed equidistantly. Okay. One thing that you have to observe here is when these beads are formed, when these nucleosomal beads are formed, you see there is a stretch of DNA that is being left over. Okay, this is a small stretch, and this is usually linked to your histone number one, H1. Okay, this is going to act as a linker. And what is the role of the linker here? Whenever we undergo overlapping, whenever the nucleosomes undergo overlapping, let's say it's getting overlapped with this. Let's say this is getting overlapped with this. Okay, so ये जब overlapping होता है, if you are not leaving a linker here, it is going to be what is it that tensile strength of your DNA will not be enough to withhold the overlapping forces, and and there can be a mechanical damage. Okay, so this way you can have your DNA cut at several spots, and you can lose your genetic content. To avoid that, what we are doing, we are leaving this gap called as linkers. So they provide mechanical strength at the same time. They provide the stability to these overlapping partners. So these overlapping partners will be now becoming a fine thread called as chromatin. Okay, so this is another level of packing. Okay, so your DNA molecule has become helical. आप एक thread लीजिए, okay, and you join it with other thread, you twist it up. So when you twist the thread, automatically the length will reduce. So that's what happens with the helix. Okay, now this helix interacts with these complexes, and uh, whatever the distance is there, that's again gone down by tenfold. Okay, and now this again interacts, and that will be forming some nanometer structures called as chromatin fibers. So when you are actually looking at a regular cell, this is the structure that you come across. This thread-like structure that you see here, this chromatin is the content that you see in your nucleus. You can't see your DNA just like this. You will be seeing your hereditary content like this. We call it as chromatin, be it a bacteria or be it a plant or be it a human being. Whatever the creature may be, you look at your DNA in this format. It's chromatin. This is called as chromatin. So once the chromatin is there, now this chromatin, when the cell has to divide, it undergoes further condensation, goes for more more folding, and that will be resulting in a complex rope-like structure called as chromatid. Okay, this is called as chromatid. So this way, uh, all of your DNA will be occupying different spaces. Okay, so this is one chromatid, this is another chromatid, and when we are doubling our chromosome, so these chromatids also get doubled up, and they will be producing these sisters. Okay, so you can see these two are same and these two are same. Okay, so whatever the content you have here, the same content will be available here. Okay, and for the reason these top two arms are called as sister chromatids, the bottom two arms are called as sister chromatids. So how does this chromatids get linked? You can see here I have indicated you some sort of a zebra crossing picture in between both the arms. Let's say this is P arm and let's say this is Q arm. P and Q has to get connected. So where do they get connected? We have a structure here called a centromere, which is usually made up of protamines, okay, and several other proteins. Because of the availability of amines and proteins, this structure is much darker compared to rest of your chromosome. Okay, so this completely makes your chromosome. So now when you are looking at a P and Q, you are actually coming across these numbers 11, 12, 13, 21, 22, 23, 24, etc., etc. Right? So what is this arrangement? This arrangement, these are nothing but these are your alleles, which are indicating some property of your body. Okay, let's say 21. 21 is indicating about the so-called hair formation and hair color. Okay, so 21 deals with all the properties of the hair. In some cases, 20. Let's say in my case, my hair is curly, it's darker. Let's say um, in some other case, somebody has a smoother hair and it's brownish in color. So this way, the properties of a particular gene, properties of a particular physical entity are depicted on these structures. Okay, and how do we identify the structure? We have a method called as chromosomal binding, and through that process, we'll be able to identify where what properties are available at what location. Okay, so these are called as alleles. These are called as alleles, and the alleles are nothing but for the same gene, you have some sort of a diversity. Okay, you observe some genetic diversity. Okay, just like the way I have given you an example, we can find different types of hairs, and that's happening because of the deviation at this gene. Okay, allele is indicative of something called as a genetic diverse pattern of some physical property. Okay, so this way we have alleles available in the nature, and the alleles are spread over on your chromosomal arms like this. What's the time?
Sir, 8:55.
8:56. Okay.
Sir,
Uh,
55, sir.
okay. Let's go through this. Okay. So now we have seen that we have a P arm and we have a Q arm. Okay. So when you look at our chromosomes in the entire body, your chromosomes are not all the chromosomes are not same. You see different types of chromosomes available, right? So if you're going to look at the different types of chromosome, you see here we have a centromere, then we have a Q arm, but there is no P arm. This is one type of chromosome. This kind of chromosome is called as telocentric. Telo, terminal end. Centromere is exactly the terminal end. Then we have acrocentric. We have two sister Qs and two sister Ps. But Ps are very, very short and they are after the centromere. So this kind of a formation is called as acrocentric. Then we have third formation called as submetacentric, where the telomere is not exactly in the center, but it's towards the center. And we have a short PM and a long QM. They are unequal. They are not very, very short nor very, very long, but they are unequal. So such kind of a formation is called as submetacentric. And we have a fourth formation called as metacentric, which means the centromere is exactly the center. We have a equal P and a equal Q. These two are same in with respect to their length and dimensions and all. Okay. So no P, telo, a very, very small P, acro, short P, submetacentric, equidistant P, metacentric. Centromere exactly in the terminal end, telo. Centromere a bit away from the terminal end, acro. Centromere a bit away from the center, submetacentric. Centromere exactly in the center, metacentric. So you can recognize the types either based on the location of your centromere and based on the availability of the PM. So we have four types: telo, acro, submeta, meta. Okay, these are the four different types of chromosomes that we come across. So function of chromosome, let's just quickly browse through. They are required for hereditary transportation of genetic content from one particular organism or a cell to another cell. That's one thing. Second thing, they control they control the entire division process. Okay, and also they control the differentiation process. Division, let's say a zygote is being formed. Zygote form होने के बाद zygote से पूरा बच्चा बाहर निकलना चाहिए. You have to get your entire organs and organ systems and all well established, right? The embryo has to develop and the baby has to develop inside the mother womb. So that all these properties are embedded onto these chromosomes. Okay, and not just that, in case of higher creatures like us, we have something called as sex determination. We have something called as male and female categories. Okay, so the male and female determinants are usually coming up from these chromosomes called as sex chromosomes. Okay, so we have 46 chromosomes. In case of humans, we have 46 chromosomes. Out of 46 chromosomes, 44 chromosomes are called as autosomes, or you can say 22 pairs are called as autosomes. Autosomes regulate all the biological characters except sexual characters. And you have two chromosomes known as allosomes. Allosomes depict only sexual characters. Okay. So in case of a male, we have a X and we have a Y. Your allosomes in case of male are X and Y, and in case of females, it's X and X. Okay. So when a baby comes out, depending upon these allosomes, you can determine the sex of the baby. And not just that, the genetic diversity is introduced by these chromosomal variations. We have a phenomenon process called as crossing over, and that actually gives you the difference. Okay. Then mutations. In as the life evolved on Earth, we have some billions and billions of creatures coming out, right? So starting from the bacteria to the other creatures, how actually these chromosomes evolved, that can be studied by looking at the mutations that are produced on these chromosomes. Okay, so these are the different functions of the chromosomes, and this is how the DNA packing is done. And with these concepts, we can move ahead with the division part. So in the next class, we'll be studying about division and all. So we stop here for today, and there are few people who joined late. I request you all to kindly answer your attendance.
Sir, 812.
Wait. Let me just open my SAP portal.
Okay, sir.
Just a second. Those who have given attendance can leave. It's already 9 o'clock. Probably the sir or madam must be waiting for you. Yeah.
Thank you, sir. Thank you, sir.
Sir,
Thank you
812.
all.
Sir,
Eight
can
one
you
two
confirm my 767?
797, sir.
seven
Sir,
six seven. I have already marked you.
835.
767. I have marked you.
Sir, we can write our roll number in chat box
Seven nine
for
seven.
giving the
नहीं
attendance.
नहीं आप बोल दीजिए ज़्यादा लोग नहीं है. 797. Okay.
835.
Sir,
8